So for figure 3, this figure focuses on the dual RNA requirements for target DNA binding and cleavage. It's made up of 5 different parts, A through E. They answer about 3 sub-questions to answer an overarching question, which is tracer RNA required for target DNA binding in order to stimulate the nuclease activity of Cas9 downstream of target recognition? So in order to answer that question, the authors start by asking a more simple and broad question, which is the entire length of the tracer RNA necessary for site-specific Cas9 catalyzed DNA cleavage? And so to answer this question, they tested Cas9 tracer RNA, CRRNA complexes, which were discussed in figure 2. And the answer to this question will be addressed in figures 3A through C, and I'll touch more on the methods later on. The next question is, what are the protospacer sequence requirements for type 2 CRISPR/Cas immunity in bacterial cells? And this question will be addressed in figure 3D. And the final sub-question I found addressed was, what specific sequence is required for efficient target cleavage? And that will be addressed in figure 3E. So for methods, they basically, for all of the different subfigures in figure 3, they use oligonucleotide DNA cleavage assays for its methods. And basically this assay uses uniquely 3' radiolabeled DNA substrates and denaturing polyacrylamide gel electrophoresis to provide evidence for the tracer RNA DNA cleaving activity of Cas9. And so this assay basically allows comparison of the effectiveness of different sequences in efficient target cleavage. And a huge advantage of this assay protocol is that it's actually quite short and it can be completed in even a couple of days. So for more specific methods of what they used for this, DNA oligonucleotides were radiolabeled by incubating with 5 units T4 polynucleotide kinase in a kinase reaction buffer. And then they inactivated it through heat. And after that, reactions were purified to remove unincorporated labeling. And then duplex substrates were generated by annealing labeled oligonucleotides with equimolar amounts of unlabeled complementary oligonucleotide, followed by slow cooling to room temperature. Cleavage products were resolved on 12% denaturing polyacrylamide gels containing urea, and then these gels were visualized, which you can see right here. So to reiterate what I was beginning to talk about in the introduction, they tested Cas9 tracer RNA, CRRNA complexes, reconstituted using full-length mature CRRNA and various truncated forms of tracer RNA lacking sequences at either the 5' or 3' ends. And you can see all the different truncated forms of these at the top of these electrophoresis images. These complexes were tested for cleavage using a short target dsDNA. So what they found was a substantially truncated version of the tracer RNA retaining nucleotides 23 to 48, which you can see right here of the native sequence, was capable of supporting robust dual RNA-guided Cas9 catalyzed DNA cleavage. And you can see that both in figure A and figure C down here. What you can also see within these figures is that truncation of the CRRNA from either end shows that Cas9 catalyzed cleavage in the presence of tracer RNA could be triggered with CRRNAs missing the 3' terminal 10 nucleotides, which you can see in figure 3B and also in figure 3C. And in contrast to this, 10 nucleotide deletion from the 5' end of CRRNA abolished DNA cleavage by Cas9. which you can see right up here. So we're next going to look at figures 3D and E. And to go more specifics into what these specifically um, are looking to do, to investigate the protospacer sequence requirements for type 2 CRISPR/Cas immunity in bacterial cells. They analyzed a series of protospacer containing plasmid DNAs harboring single nucleotide mutations for their maintenance following transformation in bacteria and their ability to be cleaved by Cas9 in vitro. So for the results they found within these, in contrast to point mutations introduced at the 5' end of the protospacer, mutations in the region close to the PAM and the Cas9 cleavage sites were not tolerated in vivo and resulted in decreased plasmid cleavage efficiency in vitro, which you can see in figure 3D. And basically this shows that the Cas9 cleavage sites and the PAM cleavage sites may be necessary for this, which I will talk about in the conclusion. And these results that they found are in agreement with a previous report of protospacer escape mutants. And then to further confirm their finding, they found results in figure 3E, which was a stretch of at least 13 base pairs between CRRNA and the target DNA site proximal to the PAM is required for efficient target cleavage. And up to 6 contiguous mismatches in the 5' terminal region of the protospacer are tolerated, which you can see in figure 3E. These findings confirm observed seed sequence requirements previously found for target nucleic acid recognition in proteins and the CRISPR complexes. So in conclusion, these figure 3 found that target recognition by Cas9 requires both a seed sequence in the cRNA, which was found in the data results from figure 3A through C, and a GG dinucleotide containing PAM sequence adjacent to the cRNA binding regions, which I pointed out in figure 3D through E in the DNA target.